Is that okay?
You might put it up a little bit.
Oh.
I would like to define it. Yeah. Can you bend it?
Check,
Bend it?
check 1, 2.
Very good. Okay. Okay, why don't we get started? So first of all, welcome everybody to the kickoff of the 2022-23 Fralin Biomedical Research Institute Pioneers in Biomedical Research Seminar Series, fondly known as PBR. It is fantastic to see people back in person. Yay. It's also fantastic to have folks online as well, but in case you're thinking about it, come on down. There's still room in the room here. So before I turn this over for the introduction, I just want to mention to everybody and remind them this is the first in this year's series, which we've been running about 11 years or so now. It's every Friday at 11 a.m. Same place, same station. I think there may be one or two Fridays throughout the year we don't have a talk, but other than that, they're pretty much regularly scheduled. So please put it in your calendar and plan to be here. You'll hear some exciting pioneers, including starting off today. And Dr. Chappell will be introducing today's speaker, which is jointly sponsored by the Center for Vascular and Heart Research. And he'll be telling you all about Dr. Hershey. But I just want to say that Dr. Hershey and I overlapped for a number of years in Houston at Baylor College of Medicine. Have known each other for many years, and I've continued to follow her career from a distance. And I'm incredibly impressed, as I'm sure you will be today. I won't say any more since Dr. Chappell is going to introduce her. John, all yours.
Great. Thanks so much, Mike. So yes, it is an absolute pleasure for me to introduce Dr. Karen Hershey as today's PBR seminar series speaker. Dr. Hershey's academic career began as an undergraduate at Penn State University, go Nittany Lions, followed by her graduate education at the University of Arizona, where she was already demonstrating her talent for science as a Calvin pre-doctoral fellow. So this is fun to get to learn about your colleagues. Like, you're already impressed, but then you read their CVs, like, man, even more impressed. Holy cow. She went on to receive an American Heart Association fund as a postdoctoral fellow at the University of Arizona, and then transitioned to a second postdoc position with Pat Moore at Harvard School of Medicine, again receiving an NIH NRSA fellowship. And it was during these years, around these years, that Dr. Hershey contributed to numerous impactful publications, one of which being my favorite, so I have to mention it, demonstrating connexin 43 activity in vascular mural cells. It's again one of my favorite in circulation, potentially pericytes maybe, and their differentiation programming through the TGF-beta pathway. So Dr. Hershey then began as assistant professor at Baylor College of Medicine, as Mike mentioned, in the Department of Pediatrics with adjunct appointments in Baylor Department of Molecular and Cellular Biology, and the Department of Bioengineering and Rice University. I think they're like their adjunct appointments as much as we do. She continued on her promotion pathway as an associate professor with tenure in 2003, and then for full professor in 2011, all at Baylor and Rice University. She then moved her lab to Yale University in 2011 and co-directed the Cardiovascular Research Center there until 2019, growing it as one of the highly respected vascular centers in the United States. And then she migrated in 2019 south to the University of Virginia to the Cell Biology Department. She wasted no time there, becoming successful, also becoming the founding director of the Developmental Genomics Center at UVA, and then also becoming the associate director of the Medical Scientist MD PhD training program as well. So Dr. Hershey has won numerous awards, has been the chair of a wide range of advisory boards and organizing committees and funding agencies. Again, looking through the CV, it was just amazed at the breadth of things that she's been involved with, as well as a chair of international conferences, again, including one of my favorites in Barcelona, Spain, where we shared a nice glass of scotch at the Gordon Conference for Endothelial Cell Phenotypes and Health and Disease. It was a fantastic time and fantastic science, including Dr. Hershey's work as well being featured. Dr. Hershey has published over 115 journal articles in top-tier journals such as Angiogenesis, Science, and Nature. As mentioned earlier, Dr. Hershey has been prolific in attracting funding, is currently a PI, co-PI, or mentor on 5 NIH-supported grants, as well as one from NASA. So I meant to ask you about that, but that's very curious. I would certainly continue to share Dr. Hershey's wide range of accolades and successes, but I wanted to offer other reasons why Dr. Hershey is one of the scientists whom I hold in such high regard, wanting to model my own career after hers. I met Karen when I was a postdoc at the University of North Carolina, and it was one of those PI talks with the trainees sessions where Karen took a genuine interest in us as trainees, our projects and our careers, sharing her experiences, both triumphs and not so much triumphs, offering encouragement in those moments to stay with it. And in her talk during her visit, I was so struck by how she presented and discussed her data giving a sense of being guided by curiosity and wonder rather than self-promotion and agenda. And I've always had the pleasure of interacting with Karen in other capacities in a similar manner, where she has always been a source of encouragement and a kindred spirit in pursuing our shared fascination with the vascular system. There's so much more I could say. I'm afraid she's turning real red, no. But I wanted to close by saying thank you, because we initially scheduled her to be one of our speakers during the onset and progression of the pandemic, and she stuck with it. I can't say that was true for every speaker we've invited, but Karen was gracious enough to come down and endure some of those delays and overcome those challenges. And it's a pleasure to have her here with us today to share with us the mysteries of an endothelial cell heterogeneity in arterial and venous specification. So without further ado, please welcome Dr. Karen Hershey.
Thank you. Thank you so much, John, for that very kind and generous introduction. And thanks, Mike, also for introducing the seminar series. I'm really happy to be here. I'm glad that we're able to do this in person. And it's really nice to see all of you in 3D. This is like my first full seminar in person, so I'm really looking forward to it. I'm also super happy to be here at this facility now that we're neighbors, not too far away. I'm looking forward to learning a lot more about what all of you are doing, and hopefully we can forge collaborations and grow research programs together. And so I look forward to interacting with all of you more as we move forward. Okay, so just as in terms of a broad introduction, I know not all of you think about vascular biology every day, and so I just want to tell you generally what my lab goals are and the system that we work in. And so we've been trying to understand the cellular and molecular basis of blood vessel formation for a number of years. And so this is an overview of the cellular processes that are thought to be involved in this process. And so this begins with the differentiation of multipotent mesodermal progenitor cells into angioblast or pre-endothelial cells, and then the formation of endothelial cells during development. And then these primordial or primitive endothelial cells become specialized into different phenotypes like arterial and venous, lymphatic, also give rise that are called hemogenic endothelial cells that give rise to all of the hematopoietic stem and progenitor cells during development that will serve as the basis for the development of the hematopoiesis system. And so endothelial cells, as they're remodeling and becoming specialized, they also recruit the cells that will form the vessel wall. And so endothelial cells put out signals that recruit mesenchymal progenitors, and then when they come into contact and they communicate through some of our favorite proteins, connexin proteins, there's activation of TGF-beta, as John mentioned, and this transforms the mesenchymal progenitors into a smooth muscle or pericyte fate. And then the vessel wall is formed through these interactions. And so over the many years that my lab has been studying this process, we initially started studying these interactions between endothelial cells and mesenchymal progenitors. And we've actually moved backwards in this process. And now we are mostly focused on trying to understand the mechanisms of endothelial cell differentiation from mesodermal progenitors, and then trying to understand how they acquire specialized phenotypes that enable their very specialized functions throughout the body. And so we do most of our studies in the mouse embryo model system. We also collaborate with zebrafish investigators as well. And then we try and understand whether the principles that drive this process in vivo could be used to direct the fate of human stem cells in culture. And so we work with both human embryonic stem cells and iPS cells as well to use the information that we gain from developmental studies, too many buttons here, that we gain from our developmental studies to drive the fates of the pluripotent human cells. And so we are able to then understand not just the mechanisms of human vascular and hematopoietic development, but then we're also able to generate these cells for collaborative projects that are more regenerative medicine based and also tissue engineering oriented. And so we do these studies and collaborations with investigators who work specifically on these projects. And we're still involved in a project trying to promote neurovascular regeneration in stroke injured patients. And this is in collaboration with investigators at Yale. And we've also been involved in this very interesting project trying to create organs on a chip And this project is headed by Gordana Vanjek-Novakovic at Columbia. And so when we started this project a number of years ago, it seemed like science fiction, but in fact, we now have multiple different kinds of organs on a chip that are connected with a vascular network, which is very cool. And so these are studies that we do in collaboration, as I mentioned, trying to apply principles that we learned from early development. And so we know the blood vasculature is an extremely important organ system because it's ubiquitously distributed throughout our bodies, and it provides oxygen and nutrients to all tissues and removes metabolic waste. And we know that during embryonic development, the proper formation of blood vessel structures is critically important for embryonic survival. But this process is also important postnatally. It's involved in tissue growth and tissue repair and in physiological situations such as pregnancy and lactation. And then there are a number of prevalent diseases where this process goes awry. And so in some cases, there is too much vessel formation. In other cases, vessel formation is stunted and leads to tissue ischemia. And this contributes to a lot of disease cases within the United States. And so we're trying to understand how blood vessels form normally so that we can contribute to trying to figure out how to clinically treat patients that have disrupted vessel formation. And so the other vascular system that is ubiquitously distributed throughout our bodies is the lymphatic vasculature. And so the lymphatic vessels collaborate very closely with the cardiovascular system to enable other important functions such as maintaining fluid homeostasis, immune surveillance, and also the uptake of dietary fats. And when this is dysregulated, it leads to different types of problems, including edema and swelling, fat malabsorption, and immune dysfunction. And so all of the vessels in the lymphatic and the cardiovascular system, they're all lined with endothelial cells. And endothelial cells have to acquire different phenotypes and different structural properties that will enable distinct functions of these vessel types. And so, for example, if we just look at arteries and veins, for example, in the blood vasculature, we know that the microenvironment within these vessels that the endothelial cells are exposed to are very different. And this in part plays a role in modifying the structural components of the vessel. So for example, on the arterial side of circulation, these vessels are exposed to very high shear stress, and the blood that they're carrying is very high oxygen and nutrient content. And so these endothelial cells within arteries are very tightly connected, and they're surrounded by multiple layers of smooth muscle cells that offer structural integrity, and they're also innervated that allow for contractility. And on the venous side, it's the opposite scenario where the blood flow in these vessels is really slow, and so it's very low shear stress, and the oxygen that's carried in venous structures is largely in most tissues low oxygen and low nutrient content. And the flow is so low that valves have to form to prevent the flow backwards in these vessel structures. And this is very similar to what's happening in lymphatic vessels, where flow is also very slow and valves form as well. And in fact, during development, lymphatic vessels form from some of the larger venous structures in the embryo. And so we are trying to understand the mechanisms that enable the endothelial cells to acquire these specialized phenotypes and enable these distinct functions. And so this is really important because sometimes this doesn't happen properly, and it leads to sometimes potentially lethal vascular malformations. And so if we look in the normal situation where the arteries are typically delivering oxygenated blood to tissues and veins are returning deoxygenated blood to the lungs and heart, it's this region in the middle in the capillaries where all of this exchange of oxygen and nutrients occurs. And sometimes when arterial-venous specification does not happen properly and capillary structures are not properly formed, we end up with what are referred to as arterial-venous or AV shunts or arterial-venous malformations. And this is actually not uncommon in the United States. And if these malformations are large and they're in critical organs, such as in the brain, the lung, in the liver, they could also be fatal. And so again, we're trying to understand the mechanisms that lead to the specification in the normal situation so that we could also try and understand the genetic mutations that lead to these vascular malformations that happen during development, and sometimes they arise postnatally as well. And right now there are no curative treatments for vascular malformations, and so we're we're trying to move in that direction. And one of the most interesting things that endothelial cells do, especially during development, is that they give rise to other cell types. And so, as I mentioned earlier, a very small subset of endothelial cells in the developing embryo acquire a hemogenic phenotype. And these hemogenic endothelial cells are then responsible for giving rise to the hematopoietic stem and progenitor cells that form the foundation of the hematopoietic system. And so endothelial cells can also give rise to mesenchymal cell types. And I know that some of you here are studying this endothelial to mesenchymal transition. And during development, this is really important, for example, for the formation of the heart, but it also can happen postnatally. And in those situations, it's not such a good thing and can contribute to pathology such as atherosclerosis. So in my lab, we were very interested in this process, trying to understand how endothelial cells acquire this hemogenic phenotype and how some of them acquire this blood-forming process or this blood-forming potential. And so we think that this molecularly is two distinct steps. The first being the induction of this hemogenic phenotype in the endothelial cells, and following that is the generation of the hematopoietic stem and progenitor cells from these hemogenic endothelial cells. And so our questions have been trying to understand what are the unique characteristics of these hemogenic cells, how is this specification happening, and are the mechanisms that regulate this specification, do they apply to other types of endothelial cell specializations? And so before I get into any details, I just want to briefly review this process of hematopoietic development in the mouse system, which is what we study. And so this is the timeline for the mouse. And the same as in all mammals, there are two waves of blood production. And the first is referred to as primitive hematopoiesis because it is the formation of only immature erythroblast and not so much myeloid and lymphoid cells. And this takes place initially in this extraembryonic yolk sac tissue, which surrounds the embryo, which is shown here. And it serves as a vascular connection between the placenta and the embryo proper in the mouse. And the next step of blood production is referred to as definitive hematopoiesis, and it's the generation of not just erythrocytes, but myeloid and lymphoid cells as well. And this takes place in different places within the embryo and extraembryonic tissues as development continues through gestation. So initially, it is also in this extraembryonic yolk sac, but a day later after primitive hematopoiesis, and then within the placenta a day later after that. And ultimately, it becomes the hematopoietic stem cells are generated in this region called the aorta gonadomesenephris region, or the AGM. And it's in this tissue where the cells, the progeny from the hemogenic endothelial cells that occur in all of these tissues, it's particularly in this region where the progeny are thought to be true hematopoietic stem cells. And that's if you derive them from this tissue and you transplant them into lethally irradiated recipients, they will repopulate all of the hematopoietic lineages, whereas the progeny that arise in the yolk sac and the placenta are thought to be more multilineage progenitors, and they don't really generate lymphoid cells very well. But all of the hematopoietic stem and progenitor cells that are formed in these tissues then migrate into the fetal liver later in gestation as this tissue begins to form. And here they propagate and they start to differentiate, and ultimately they migrate into fetal bone marrow, which is established pretty late in gestation. And in humans, then the hematopoietic stem and progenitor cells that are generated in these tissues reside predominantly within bone marrow. In mice, there's also postnatal hematopoiesis within the fetal liver. But this is just to show you the importance of getting this process right during development. So this process of hemogenic endothelial specification and enabling their ability to create these hematopoietic stem and progenitor cells really has an impact on postnatal hematopoiesis and can cause adult phenotypes.
In mice, does the liver provide a source of hematopoiesis throughout life?
I don't think throughout life, but initially postnatally. So at least within during the weaning period. So there's hematopoiesis from liver. So in humans, it's not so much. It's pretty much restricted to bone marrow, except in cases of bone disease. And then there is hematopoiesis within the liver and the spleen and extramedullary hematopoiesis. Okay, and so, So we wanted to be able to watch this process happening in vivo. And so when I was at Baylor, we worked very closely with Mary Dickinson to create these transgenic animals so that we could watch this process of blood generation from the endothelium in live tissues. And so this is imaging in the yolk sac, and I'll show you the next is an image of an AGM explant. So in these animals, the endothelial cells are labeled with the FLIC promoter, and this is the endothelial cell enhancer region of the FLIC promoter driving M-cherry, and blood cells are labeled with LMO2, which is a transcriptional regulator required for hematopoiesis, and in this case, the promoter is driving GFP expression. And so if you watch this movie, it's going to play, we could see the red endothelial cells giving rise to green blood cells, for example, here. And then it pops into circulation. There's going to be another one over here. There we go. So you can see the green cell being generated from the red endothelial cell, and it pops immediately into circulation. And this is the way that it happens within the yolk sac tissue. And this is actually different than the way it happens in the AGM. So this is a cross-section of the aorta. And again, the endothelial cells are labeled with M-cherry, but in this case, the blood cells are labeled with epsilon globin driving GFP. And so if you watch in this region, you can see again, the endothelial cells generated the green blood cells, but the process is a little different. So you could see the green blood cell, and then it divides and it forms. Eventually, the movie's going to stop, but it forms what's called an intra-aortic cluster in the aorta wall. And so it stays there, and the progeny, it's thought that it's this process that makes it different from the yolk sac, and it determines the outcome of the progeny that are generated from the hemogenic endothelial cells, and that there is molecular signaling within this cluster that is enabling the multipotency and the stem cell characteristics, as opposed to in the yolk sac, where the cells do not become true stem cells and they don't have lymphoid potential. So we and a number of other labs are very interested in understanding this microenvironment and how it dictates the fate of the progeny generated from the hemogenic endothelium, because at this point we can't really see from studies that we've done so far large molecular differences between the hemogenic endothelial cells in the AGM and the yolk sac, although with single cell analysis and other kinds of more advanced techniques that we're using now, I'm sure that we will unearth molecular differences between these cell types. Okay. So we spent a lot of time and a lot of money in the flow core trying to understand the phenotype of these specialized endothelial cells that have blood-forming property. And so just to make a long story short, what we found is that they have these characteristics. So they express the VEGF receptor 2 or FLIC1, and they also express c-KIT, which is the receptor for stem cell factor. And they are not expressing any cell surface proteins that would be expressed by cells that are committed to hematopoietic lineages. And they also have this SP phenotype, which our colleague Peggy Goodell found that is a characteristic of hematopoietic stem cells in adult bone marrow. And this is, the cells are called SP or side population because they overexpress multi-drug resistant pumps that enable them to extrude small molecules, including DNA binding dyes that they extrude. And so you can use this characteristic in flow cytometry to identify all the cells that overexpress these pumps and have this side population or SP phenotype. And so it's just interesting that we did our studies early in collaboration with Peggy's lab to show that these hemogenic endothelial cells have the same property as the hematopoietic stem cells do in adult bone marrow. And it's thought to confer some protection of these multipotent cells from differentiating factors. But I think that's still under investigation. But nonetheless, so if we isolate endothelial cells that have these characteristics, either from the yolk sac or the AGM, and we plate them as single cells, we can see that they have a morphology that's similar to endothelial cells. They have functional processes that are similar, like the uptake of diacetylated LDL. They also express eNOS, which is not shown here. And if you culture them on fibronectin, they will form a little endothelial cell colony. And then if you expose them to methylcellulose, which is a substance that contains a lot of growth factors and soluble factors that promote blood formation, you can see this explosion of blood forming potential from single endothelial cells. And so we get this generation of erythroid colonies and myeloid colonies, granulocytes and macrophages, and importantly, these colonies that have mixed phenotypes. granulocytes, erythrocytes, monocytes, and macrophages. So a single endothelial cell can give rise to this multi-lineage hematopoietic colony. And this was part of the proof that we had to demonstrate that these are in fact the right phenotype of the hemogenic endothelial cells that give rise to multi-lineage progenitors. And so it was really important to define, oh, John, go ahead.
I was curious, do they respond to different flow conditions? I'm just curious because I imagine the source where they're coming from, they're under different mechanical load or shear stress forces. I just wonder if the HGM cells would maybe align more quickly or readily. Right?
Yeah, we haven't. That's a really good question. So we haven't done any flow studies. So the number of cells that are derived from these tissues is really low. And so we haven't been able to really get enough numbers to do studies in vitro in cultured cells. So we've been mostly studying their function just really as single cells and then following their behavior in vivo. Rob?
Yeah, sorry to disrupt the flow, Karen. Let's go. It's wonderful. But so the endothelium, have any lineage or clonal studies been done on it to demonstrate whether the endothelium is a uniform field with respect to its ability to generate these cells, or is there some sort of specification within the endothelium, even at this earliest stage?
Okay, so I think I might not have heard all of that, but I think the question was, has lineage tracing being done to show that endothelial cells give rise to blood cells? And I'm sorry.
Are the hemogenic endothelial cell a subpopulation in the endothelium that sort of have somehow differentiated from other endothelial cells, say a neighboring cell? Are they as equally likely to contribute to the hemogenic property?
So I'm sorry, I'm still not hearing you very well. Maybe.
Yeah, are the neighboring cells, is there heterogeneity or can any endothelial cell?
Yes, basically.
Yeah.
Can any endothelial cell give rise to these cells, or is there like a specified?
So in the yolk sac, it seems to be not so specific, but in the AGM, there are specific regions where these hemogenic endothelial cells form. And then there's a limit to how much endothelium within the aortic lumen, how broadly this expands. And so we found that some of the regulators, there are microRNAs that regulate, the spatial distribution of the hemogenic endothelial cells. Lineage tracing has been done, but it's just using V cadherin in general and not subpopulations of endothelial cells to look at.
I guess the way you could also ask the question is there an intermediate progenitor type that has to be specified from a differentiated endothelial cell, or does it just go endothelial cell, hematopoietic progenitor?
Yeah, so there definitely are intermediate phenotypes that could be analyzed. Okay, thank you for that interpretation. So there are definitely intermediate phenotypes between endothelial cells and bona fide hematopoietic stem cells and progenitor cells. And these have been identified largely through single cell RNA-seq analysis. So Nancy Speck and other labs have done this, and they have proposed these intermediate phenotypes. I mean, localizing them and watching them in vivo, I have not seen that so far. But just by doing bioinformatic analysis and related studies, I think there definitely are intermediates. I don't know the stability of intermediates. And this process is actually interesting because there's like a time limit. So in the yolk sac, it starts to happen with about embryonic day 8.5, but then it shuts off at 11.5. And in the AGM, it starts maybe around E9, and then it shuts off like when the fetal liver kicks in and is more hematopoietic. And so there are these waves that are going on in different tissues throughout gestation, and we still don't really know what's the on-off switch here. And I'll show you later that retinoic acid is an important promoter of this. Yes, you're one of your friends. And so, it could be that setting up the gradient of retinoic acid signaling is really important for allowing the induction in specific regions at specific times.
Niche in the small blood vessels, because they gather into a niche in bone marrow where they're in a place where they have all, we presume, everything they need to maintain their multipotency and also to progress through the lineage. but in the vessel itself, there's not a domain where they get particular.
There is in the aorta. Yes, there is. And then its position and its size is definitely regulated. And we found some microRNAs that regulate, that if they're absent, then they go ectopically where they're not usually. Okay, so it is regulated, but it's all of the regulators are not known. Okay, and so we did a lot of studies to try and understand the molecular regulation of this hemogenic specification. And I'm not, in the interest of time, I'm not going to go through all of these studies, but I just wanted to point out that retinoic acid is a very important promoter of this process. And there are a number of downstream factors that are important in the process. And so we knew that retinoic acid could potentially be involved in hemogenic specification because we found a number of years ago now, when I look at the dates, I'm like, wow, I'm really old. But so we found that retinoic acid signaling is really important for vascular development in general, because in these mutants that lack retinaldehyde dehydrogenase 2, which is the enzyme that's needed to convert vitamin A from that the embryos get from maternal circulation into active retinoic acid. If the embryos are lacking this enzyme, they are essentially retinoic acid deficient. So in these animals that end up with a lot of developmental problems, but initially early on, one of the things that's really messed up is vascular development. So they form a vascular plexus, and this is the yolk sac tissue shown here, but it never remodels into an arterial-venous circulatory system. And we found that one of the basic problems with the vascular endothelial cells is that they are hyperproliferative, which I'm going to come back to. And so in this case, the endothelial cells that are retinoic acid deficient, they don't upregulate cell cycle inhibitors such as P21 and P27, and they spend less time arrested in G1 state of cell cycle and more time actively proliferating. And we found also in these animals, if we isolate the hemogenic endothelial cells that we now have a cellular definition of, and we look at the development of those and also the multipotent progenitors that they produce, we could see that this is also significantly impaired in the retinoic acid deficient embryos. And interestingly, this phenotype could be rescued because you can add or give back exogenous active retinoic acid to the embryos either through maternal food during gestation, or you can take the embryos out and you can culture them in the presence of active retinoic acid. And we can completely rescue the development of hemogenic endothelium and these multilineage progenitors, which was suggesting retinoic acid is critically important for this process. And then we went on in a number of other studies to demonstrate that retinoic acid, and this is a model of what's happening in the yolk sac tissue shown on cross-section. And we know that retinoic acid is produced in this visceral endoderm layer, and it signals below to the mesodermal progenitors where endothelial cells are forming. And some of the endothelial cells are highly expressing retinoic acid receptors, alpha 1 and 2. And this retinoic acid signaling leads to the upregulation of c-KIT, which I mentioned earlier is the receptor for stem cell factor. And downstream of this is activation of Notch signaling, which leads to a cell cycle arrest that is driven by expression of P27, which is the CDK inhibitor. We found that this cell cycle arrest is really critically important to enable the upregulation of other factors, such as this transcriptional regulator, RUNX1, that Nancy Speck and others have shown is a critical factor needed for the generation of hematopoietic stem and progenitor cells from these hemogenic endothelial cells. And then a number of other lab groups have shown that this process of hematopoietic stem and progenitor cell development is very complicated, and it involves a number of signaling pathways that have been independently identified, and we're still trying to figure out how signaling is coordinated. But as vascular biologists, we were most interested in this part of the process. So what is going on, and how is P27 and cell cycle control regulating hemogenic specification? And so I just want to show you this one piece of data from this studies that really we were surprised by. And so in these studies, we're isolating endothelial cells from yolk sac tissues at E8.5 when hemogenic endothelial cell development and definitive hematopoiesis is happening in that tissue. And so we isolate the cells from wild type and also these retinoic acid deficient mutants. And if we look at the cell cycle distribution of the endothelial cells, we could see that it is messed up in the mutant, as I mentioned earlier. And so there are more cells that are actively cycling in the mutant and less that are growth arrested. And then associated with this lack of cell cycle control or dysregulated cell cycle control is impaired hemogenic endothelial cell development. And then if we re-express P27, which is downregulated in these mutants, back to wild type levels, we can rescue the cell cycle defect, which is not that surprising, but we also can rescue hemogenic endothelial cell development, which was really surprising to us because we're like, how is this happening? And just to remind you what this protein does, it's a member of the KIP-CYP family along with P21 and P57. And so these proteins interact with CDKs in G1 state of cell cycle, and their interactions then prevent them from interacting with other proteins such as cyclins and other proteins that are not shown here in this very simplistic model of cell cycle control. And collectively, when these proteins are functioning, they prevent cells from moving from G1 into S phase. And so they are stuck at this G1 to S restriction point. And in retinoic acid deficient endothelial cells, where there is downregulation of these inhibitors, then the cells continue to cycle and they are less arrested in this G1 state. And so we wanted to know, is this true in vivo? Are the hemogenic endothelial cells really in a different cell cycle state than other endothelial cells? And so to get at this, we adopted these mice that are referred to as Fuchi mice, and this is their abbreviation up here. And I know that some of you are using these mice, are familiar with them, and they take advantage of the dynamic control of two cell cycle proteins. And so there are two fusion proteins that are expressed by these mice. This is a part of the human geminin gene driven by M. venus, and then the human CDT1, a portion of that driven by M. cherry. And so when cells first divide, neither of these reporters are expressed, and this is referred to as early G1 state, and the cell nuclei are not labeled, they're not fluorescent. But as the cells progress in G1 state, the CDT protein and the M. cherry fluorescent protein accumulate and the nuclei become red. If cells transition into S phase, they are temporarily expressing both reporters, but in S phase, CDT1 protein and M. cherry are quickly degraded, and then during SG2M, M. venus and geminin protein accumulate and the nuclei are green. So you can use this fluorescent reporter to look at cell cycle state changes in imaging, which would be cool to do in the retina. But also we could use flow cytometry, as we've done in these studies, where we can isolate endothelial cells from this AGM region at E10.5 when definitive hematopoiesis is happening in that tissue. And we can use flow cytometry to isolate endothelial cells in these different states of cell cycle. And then we can quantify this. And what we found, in fact, is that the endothelial cells that are hemogenic in nature are, in fact, have almost all of them are arrested in this late G1 state relative to endothelial cells that do not have blood forming potential. And so this was interesting to us because a number of years ago, stem cell biologists have identified cell cycle state as windows of commitment. And they specifically found that G1 phase is a time when there's differential expression of transcriptional regulators, changes in chromatin structure, and so forth. And this is a phase for embryonic stem cells that allows the cells to undergo state change. So, and it matters where you are in G1. If you're early in G1 state, and embryonic stem cells are able to receive signals that enable differentiation of mesoderm and endoderm. If you're later in this G1 state, the embryonic stem cells respond to ectodermal differentiating signals. And so what we had found in our studies is that this G1 arrest is necessary for hemogenic specification, but it's not sufficient. So if you arrest the cells, but you don't have the proper signals nearby, you don't undergo this phenotypic change. And so we're still trying to understand what factors actually promote the hemogenic phenotype in this G1 state. But we're also very interested in understanding if this applies to other endothelial cell specializations. So is this G1 arrest required for other types of specialization? And so I sort of showed you that this is the pathway that leads to hemogenic specification. So it's initiated by retinoic acid, but ultimately it leads to Notch activation and G1 arrest driven by P27. And a number of other lab groups have shown that some of these same players are very important for arterial specification and lymphatic endothelial cell specification. And so our hypothesis was that this is a common mechanism, so that Notch signaling is required for cell cycle arrest to enable all of these endothelial cell specifications. And so what we are studying now is trying to understand, is this true for arterial venous specification and lymphatic specification? And so I'm just going to show you briefly some studies that we've done on arterial fate. And so in these studies, we adopted a different model, and this is postnatal retinal vascularization model, which some of you are familiar with. And so this is this tissue is interesting because in rodents, the retina is vascularized postnatally. And so we could look at this tissue in flat mount, and we could look at different stages of vascular development, plexus formation, and then arterial venous specification over time in different postnatal days. So initially, when this is postnatal day 0, there is hardly any vasculature present in the superficial layer of the retina, and then it spreads out radially to cover this tissue. And at different stages, you can see there's plexus formation and then arterial venous specification. So it's a really nice tissue to monitor arterial venous specification and remodeling. And a number of labs are using this tissue for studies like this. And so we wanted to know, what's the distribution of cells that are proliferating in this tissue? And so is this higher in cells that are less differentiated? And that was our hypothesis. And that's in fact what we found, is that the endothelial cells, by using phosphohistone 3 staining, which labels cells undergoing mitosis, we could see that the cells that are actively proliferating are in this region that is still a primitive plexus and has not undergone arterial venous specification. And so the endothelial cell proliferation is highest at this vascular front, and it's suppressed as arterial and venous specification is occurring. But the question was, okay, how is endothelial cell proliferation suppressed during this remodeling process? And is it actually required for arterial venous specification? So to make a long story short, the answer is yes. Okay, and so Jen Feng, when she was a postdoc in my lab, did these studies to show that in this developing retinal vasculature, one of the important drivers of arterial specification and cell cycle arrest is shear stress. So in these studies, she found that shear stress, particularly at magnitudes that are experienced by arteries and arterioles, this maximally activates Notch signaling. And this leads to an upregulation of connexin 37, which we believe is a transcriptional regulation. And downstream of connexin 37 expression is cell cycle arrest that's driven in a P27 dependent manner. And so we're still trying to figure out how connexin 37 protein is leading to P27 upregulation. And we have some data about that, but it's not solid yet. But we do know that P27 is required downstream of connexin 37 to cause endothelial cell cycle arrest. And that if this does not happen, the endothelial cells do not undergo endothelial cell arterial specification.
Yeah. So what's the transducer of the physical stress that activates Notch signaling? And is it activating additional expression of Notch on the cell surface? Is it activating the cleavage of Notch from an existing pool?
Yeah, great questions. And so we were doing these studies in collaboration with Martin Schwartz when we were at Yale. And so we still don't know the answer, but he had identified a mechanosensor complex that included PCAM, V-cadherin, and VEGF receptor 2 earlier in his career. And he doesn't think that this is happening through the mechanosensor because the timing isn't aligning with his previous studies. Other lab groups have shown that mechanical forces themselves can change the conformation of the Notch receptors and allow for cleavage of the intracellular domain. So we think that's what's happening, but Martin's lab wanted to study that in more detail, so we have not specifically followed up on that. But we do know the shear stress leads to increased cleavage.
Sure. So it's been well known for a long time that there, in the developing retina, there are these waves of activity and calcium signals, all the glial cells, neurons, et cetera, spreading across the retina. And
it occurs to me as you're forming these AD complexes that there might be a relationship in that. So my question is, A, have you either looked at the data where people have shown the patterns of these ways of activity, overlaid it with this, or done the combined experiment? And the second part of that is, I guess it relates to these questions. I was trying to understand that what I saw in the spreading out of the vasculature of the retina was following the growth of the retina. Was it just, or was there potential shear forces generated by differential movement of the whole retina and just the vascular tissue?
Yeah, I mean, those are all really great questions. And so in terms of calcium signaling, we specifically have not looked at that. Definitely the vasculature is growing as the neuronal cell types. And actually the neural progenitor cells that are forming the retinal tissue are actually excreting a lot of VEGF, and that's sort of leading the migration and the proliferation of the endothelial cells outward in the retinal tissue. And that was shown a number of years ago by Ellie Cachette's group and some other groups. So we know that those interactions are extremely important for vascular development and vascularization of the retinal tissue. So I haven't really thought about the interactive forces between the cell types and, you know, how the patterning might affect. I mean, it's a really interesting question. And maybe the interactions also, we're trying to figure out how cell cycle state is set. And I'll show you that that's different in this retinal tissue. And so it may be that these interactions and microenvironmental factors that are between them are causing cell cycle state to change, and that enables different specifications. Rob?
Yeah, I know Jan Bird was very much working on Connexin 37 and this relation to cell cycle. And these are probably experiments that you're doing at the moment that are published, etc. But do you know whether the Connexin 37 effect is channel dependent or independent? Is for example, C-terminus sufficient or? Yeah.
So we know, so we did a lot of studies with the mutants, actually. So we have the channel dead Connexin 37 mutant, lacks the Connexin 30, the C-terminus. And then we also looked at the cytoplasmic tail itself, independent of the connexin protein. And so we think that channel function is necessary, and it involves protein-protein interactions with the C-terminus. And so I can show you some of the data that we have so far trying to understand that. It's a complicated process that we don't have completely nailed down yet, but we know that the cytoplasmic tail of connexin 37 is involved, and channel function seems to be important as well. Okay, so then what we were interested in is, okay, so what is causing the cell cycle arrest to enable the arterial specification, and what about venous specification? Is cell cycle state also important in that process? And so to get at that, we went back to these Fuchi mice, and we first wanted to see, okay, are endothelial cells in different blood vessel types in this developing retina, are they in different cell cycle states? And so we did these flat mounts of P6 and also P15 retinas. And at this point, the vasculature, the superficial plexus is established, and remodeling is not thought to be continuing. But early on, there's still a lot of remodeling and a lot of cells that are undergoing mitosis. And so what we found is, even at P6, it seems like the endothelial cells that are in arteries are predominantly in this late G1 state, whereas on the venous side of circulation, they are in this earlier G1 state. And this holds true and becomes more apparent at P15, where we could see that in the arterial side of the vasculature, the endothelial cells are really enriched in this late G1 state, and those on the venous side are in this earlier G1 state. And this is actually true in the adult. If we look at the aorta and the vena cava in these mice, we could see that the same trends hold true. Whereas in the aorta, most of the endothelial cells are in late G1, whereas those in the vena cava are in early G1. And so we can look at this a different way, and isolating the endothelial cells from the retinal tissue from these mice, we could then do flow cytometry and look at gene expression profiles. And when we do that, we see that, in fact, if we sort the cells into those that are in SG2M or early G1 or late G1, and we look at the expression of venous versus arterial genes, we could see already at P6 that there is a strong enrichment of arterial genes in the endothelial cells that are in this late G1 state, and starting to accumulate venous enriched genes in early G1, And then this is more dramatically seen at P15, where venous enriched genes tend to be expressed in early G1 as opposed to arterial genes in late G1. And so what we were thinking is, okay, so in these retinal tissues, especially the ones that have undergone remodeling and are no longer going through active cycling, the endothelial cells are growth suppressed. But there's a difference. And so on the venous side, it seems that most of the endothelial cells are in this early G1 state, whereas in the arterial side, they're in this late G1 state. And this was really interesting to us because in yeast biology, there is a known nutrient sensing restriction point between early and late G1. And so if yeast are in a low oxygen, low nutrient environment, they don't progress into late G1. And this is interesting because on the venous side of circulation, I had mentioned earlier, the blood the blood that is transported through veins is very low oxygen, very low nutrient content. So it could be that the blood composition and flow within the venous side is helping to set the cell cycle state of these endothelial cells. And also, the stem cell biologists, again, a number of years ago, have found that these states are molecularly different. And so in this case, where we have early G1 state is predominantly on the venous side and late G1 state on arterial side, stem cell biologists have already found that there are significant differences in transcription factor expression and loading, epigenetic changes, chromosome organization, and these are very molecularly different, at least in embryonic stem cells that are in these distinct states of G1. And so we wanted to know what are the molecular differences between endothelial cells in early versus late G1, and does early versus late G1 determine the propensity for endothelial cells to undergo the specification to these different phenotypes. And so to get at this initially, we did bulk RNA sequencing of endothelial cells from these P6 retinal endothelial cells from the Fuchi mice, and we could separate them into these distinct cell cycle states. And you can see that there are lots of differences in gene expression. Among them are differences in genes that regulate mitosis, which is not surprising. But we found also that in endothelial cells that are in this early G1, they seem to be upregulated for BMP signaling, and TGF-beta signaling seems to be enhanced in endothelial cells in late G1. And this was really interesting to us because other people in the field, like Sarah Duvall, has shown that BMP signaling actually drives venous specification. And it's the venous endothelial cells that are predominantly in this early G1 state. And cell culture studies have shown TGF-beta to increase the expression of arterial endothelial cell genes. And so we wanted to know is, first of all, is BMP signaling really enriched in venous endothelial cells? And is TGF-beta enriched in arterial endothelial cells? And so to get at this, we did single cell RNA sequencing of the retinal endothelial cells from wild type P6 and P15 mice. And so when we do these analysis, we could use programs like Seurat to separate endothelial cells into those that have like phenotypes and those that are just similar from each other. And then we could identify what these clusters of endothelial cells are based on their gene expression profiles. And then we could look at the expression of TGF-beta and BMP signaling in those that tend to have an arterial versus a venous phenotype. And what we see that, in fact, that the endothelial cells that have more of an arterial phenotype tend to be enriched for expression of components of the TGF-beta signaling pathway, whereas those that are in early, those that have a venous phenotype less express BMP signaling, those that are arterial less express BMP signaling, and are the venous and venous capillary are more enriched for BMP signaling. And then to understand whether or not BMP signaling actually drives a venous fate in early G1 and whether TGF-beta can drive an arterial fate, specifically in late G1, we developed these, we developed these HUVECs, human umbilical vein endothelial cells, that express this Fuchi construct. And you could, we're supposed to be a movie. Oh, maybe I have to do this. Okay, so you can see these cells in culture as they divide, they are expressing, dynamically expressing these different reporters. And And then if we isolate these endothelial cells and we separate them into those that are in early G1 versus late G1, and we treat them with either TGF-beta or BMP for 8 hours. And importantly, this is a time frame in which neither of these factors affects endothelial cell cycle state. And when we do that, we can see that in fact, the endothelial cells that are in late G1 are responsive to TGF-beta signaling, and they upregulate these arterial genes. And then in contrast, the endothelial cells that are specifically in early G1 are responsive to BMP signaling, and they upregulate these venous genes. And so what we think is going on is that these different cell cycle states are providing different windows of opportunity for the endothelial cells to respond to extrinsic signals in their microenvironment. So we think that in the venous, and when endothelial cells are in early G1, they are more responsive to BMP signaling, and this promotes a venous fate. And those that are in late G1 are more responsive to TGF-beta signaling, which promotes an arterial fate. And we're still trying to understand the molecular mechanisms, the molecular differences between endothelial cells in these different states, trying to understand how they get arrested in early versus late G1 and what's maintaining that. It looks like postnatally. And then also we're interested in understanding whether disrupted endothelial cell cycle plays a role in vascular diseases. And I don't want to run too far over, but I just want to tell you about some studies that we're doing trying to understand the role of endothelial cell cycle control in arterial-venous malformations, which I mentioned earlier is associated with lack of venous and arterial identity in these tissues. And so this process of arterial-venous malformation has long been known to be associated with endothelial cell hyperproliferation, but it wasn't really known whether dysregulated endothelial cell cycle control was actually causing these AVMs. And so this is just to show you how the distribution of AVMs in specific diseases such as in HHT, which I can actually never say that word, but we know that this process of HHT, this disease, is driven largely by mutations in the BMP9 and 10 signaling pathway. And so these ligands activate these receptors and lead to downstream SMAD signaling. And so we could see that patients that have different forms of HHT have different genetic mutations in these pathways. And so we could use this information to model AVMs in our mouse models, and we could postnatally induce endothelial specific deletion of genes such as ALK1 receptor. And we could also use neutralizing antibodies against BMP 9 and 10 to promote this pathway and AVMs in mice. And so Gail Genet, a research assistant professor in my lab, has been doing these studies, and he's done them in postnatal retina, where he has injected these BMP 9, 10 neutralizing antibodies at postnatal day 2 or 4, and then treated these animals with CDK inhibitors. And so this particular inhibitor is FDA approved already for cancer therapies. And so we wanted to see if it could prevent or rescue AVMs in this situation. And so what we found is that this is just the vasculature and control conditions. And when antibodies are provided postnatally, you can see this hypervascular plexus in the retina and also these AV shunts that form. But when we provide this, we treat with this CDK inhibitor, we can actually prevent these vascular malformations. We don't get AV shunts, and we have a less endothelial cell hyperproliferation. And within the time frame and the dosage that we're using, we don't see any significant effects on the wild type tissue. And so we can quantify these changes. You can see that there's shunts, numbers of shunts are increased with this BMP 9/10 antibody treatment, and this goes away with the CDK inhibitor. Same is true, vascular density is increased when we provide the neutralizing antibodies, and this can be corrected with this cell cycle inhibitor. He also has done this in a genetic model where he has deleted ALK1 receptor using the V-cadherin-Cre, and you can see similarly that there are AV shunts in the animals that are lacking ALK1 driven specifically in endothelial cells in these tissues, and that we actually can prevent the formation of these arterial venous malformations with this CDK inhibitor, which looks very promising. So in summary, what we found that is induction of G1 arrested endothelial cells it appears to prevent these AVMs in animal models of HHT. We are now looking at later time points and trying to understand if they could be used to treat and to regress AVMs. And so we are trying to quantify the cell cycle state changes in the Fuchi mice that have induced HHT, looking at arterial-venous phenotypes, and also determining the mechanisms by which cell cycle arrest may prevent AVMs, and whether or not we could use these inhibitors in more preclinical models to treat or regress AVMs. I think that's it. I just want to thank everybody in my lab, especially Nick and Gail and Jen, for their work on the arterial-venous specification studies. So, thank you.
Yeah. My mic. Good. Okay. All right. So, Carl, you saw your hand
First, yeah.
Thanks for the talk. I'm very interested on G1 phase of the cell cycle. So you talk about early phase and late phase. It's not clear in the phase of the cell cycle, that one in particular is very variable. Yeah. So how do you define your early and your late? And I noticed that you don't use cyclings for that, which is kind of the hallmark for measuring time in G1 phase. And the other thing that I noticed is that in your RNA-seq experiment, you have the arterial differentiation and the cyclin B1, B2 expression at the same time, which is opposite of what you actually propose in your cartoon. That is, the arterial differentiation happens in the late part of the gene.
Yeah, I mean, just to answer your second part first. So that was RNA expression. And so we have not done detailed protein expression, protein phosphorylation studies, which we are trying to do now and trying to understand more molecularly what do these early G1 states and late G1 states as read out by this Fuchi reporter, what does that really mean in vivo? So as you can imagine, we had a lot of issues with that with reviewers, specifically cell cycle reviewers who are like, we don't understand this. How can cells be in early G1 in vivo for an extended period of time? Because the current thinking is that early G1 is very short and it just goes, but that's not what we're finding. And we've done, I can't tell you, countless validation studies of this reporter, and we can see that the mRNA, the message levels for the reporters, it's not like it's repressed and it doesn't get expressed. It's just that, you know, that their proteins or the fluorescent proteins are not getting expressed. So there's something different molecularly that's happening that hadn't been previously appreciated until this report is more frequently used. And, you know, there's still a lot of controversy about what that is. And so in our paper, we call it negative and not early G1. Okay.
So try to sort out the patterning aspect of building vessels from endothelial cells versus the local hematopoietic mechanism. So particularly for the venous malformations, you think that the BMPs and the TGF betas are favoring more or less mesenchymal epithelial transition or the capacity of a dividing hematopoietic or a hematopoietic biased cell in the vascular wall to actually pop out and become a blood cell? Or is it that you're just getting a disruption of the actual tubular patterning via that mechanism?
So let me just clarify. So in the AVMs, we don't have a hematopoietic cell problem associated with that, because these are things that happen postnatally, and all of the processes of hemogenic endothelial cell development and they're giving rise to the progenitors is early in development. Yeah.
There's no interaction between BMP signaling and the Notch signaling for actual.
In hematopoietic development from endothelium layer, no, not that we are aware of.
The BMP TGF beta balance is it a patterning mechanism, or is it a just hyperproliferation? Or
I don't really know, and I think that's we have to sort that out. So
it goes to Mike's question, because there are other ligands that can be regulated, or actually BMP sources could be coming from retinal cells cells.
Yes, definitely. And so we haven't done a thorough evaluation of that, and we are like working toward, as you can imagine, it's pretty complex to figure that out. But we haven't looked at the gradients of the protein expression of BMP versus TGF beta. We did look at signaling components. The expression of receptors and ligands is different in these different molecular states. In the endothelial cells, we haven't looked at other cell types to see if there's coordinated like ligand expression on one side versus the other from other cell types.
Which could interact with
I think we had two more questions, maybe here up front and maybe Sammy in the back. So maybe from
Thank you.
You mentioned that the 5 ST cells, combine that when there's some excitement about the ST cells empowerment status. Do you think this will be relevant in the heart?
Well, that has been a favorite hypothesis of ours, but we have not proven it yet. And so I don't know that it happens. You know, sometimes I will suggest that to hematopoietic people and they're like, no, that doesn't happen. But yeah, I don't know.
Sammy, last question.
Yeah, just quick question. I was just wondering if you know if BMP9, 10, or ARC1 regulates connexin 37 expression, localization, or function?
We haven't tested that directly. So whether BMP9 and 10, that signaling pathway regulates connexin 37. So we haven't specifically looked at that. We know that connexin 37 has to be upregulated specifically to enable arterial specification, but it is expressed in venous endothelial cells as well. But I'm not sure if that's a driver. We know Notch is a driver, but not sure about other drivers.
Well, thank you again for such a fantastic
All right,
talk. Let's thank
thank
Dr.
you.
Hershey.